Hello everyone, this is Dr Bishal Gyawali from Queen's University, Kingston, Canada, and welcome back to the Skills Lab series in Medscape. So today we are continuing with how to read a phase 3 clinical trial report. As you may have watched in the previous 4 videos, we talked about some background information even before we start reading the clinical trial report. And in last video, I talked about abstract, when to read the abstract, and why to read the abstract. So today, continuing with that same series of how to read a phase 3 clinical trial report, what comes after abstract is introduction. So today, let's talk about what to look out for in the introduction. So introduction is the section that sets the background for why the trial was done. So there are just a couple of things that I want to look in the introduction section. One is the biological rationale for the drug, especially if it's a new drug. I want to know what the biological rationale was for doing this trial. Does it make sense biologically? And the second one, and the most important thing that I want to look for in the introduction, especially for a phase 3 clinical trial, is has a phase 1 or phase 2 trial been done in the past for the same drug, ideally in the same setting. And this is important because we don't want to see a phase 3 trial done without a prior phase 1 or a phase 2. And we briefly talked about different phases of clinical trials in the I think first video, but the role of phase 1 trial is to establish safety, and I'd assume that almost all the new cancer drug trials will have undergone a phase 1, but phase 2 is tricky. Nowadays, we have seen several trials jump from phase 1 directly to phase 3 without undergoing a phase 2. And why does that matter? If we look at data of 100 preclinical studies that happen, only 5% go on to receive approval. And the biggest risk of failure would be at a phase 3. And the biggest investment from drug development point of view also is at phase 3 part of the trial in the drug development process. A typical phase 3 would cost somewhere around $22 million to conduct, whereas a phase 2 would cost around $11 million to conduct. So we don't want, ideally, a drug to fail at phase 3. We want better screening at phase 2 so that drugs that are more likely to be successful at a phase 3 are screened from phase 2. So there have been a couple of studies looking into this. So one of these studies showed that in a phase 2 trial, the objective response that we see in a phase 2 seems to be a useful endpoint for screening new targeted agents because it predicts for eventual success in phase 3. So that is the role of the objective response rate in a phase 2 trial, actually, whether or not that drug will be successful at phase 3 rather than approving drugs based on phase 2 response rate itself. And similarly, in one of the analysis that we did about the phase 3 trials that failed, we saw that for 42% of the trials that failed at phase 3, a phase 2 trial was not available. And of those trials where a phase 2 was available, 28% of them had actually failed in phase 2. And despite the drug failing at phase 2, it was still tested in a phase 3, and of course that phase 3 failed as well. So for every phase 3 trial in the introduction, we want to check whether or not a previous phase 2 was done and whether that phase 2 was successful, or the phase 3 trial was conducted despite the phase 2 failing, in which case we would assume that the phase 3 was done just to chase a statistical significance and get the drug approved without it passing through the phase 2. So that's all about introduction. Then we want to move on to the methods section. And in methods section, we'll see different subsections that are reported. And as I keep saying, methods is the section that is the most important to understand the trial, but is the least read section. So I want to encourage all of you to actually go through method section in detail. So I'll tell you what to look into the method section. The first paragraph of a method section ideally talks about what type of trial, what type of patients were included. That means the inclusion and the exclusion criteria in the trial. And this is very important because we want to make sure that the patients who are enrolled in the trial are representative of patients that we see in the daily clinic. That means we want to see diversity in patients that are enrolled in these clinical trials. For example, how many, in terms of gender diversity, male, female participation, in terms of age diversity, whether elderly patients included, in terms of race and ethnicity diversity, what ethnicity or race of patients have been included. In terms of diversity in performance status, are patients with performance status of 2 included? So we want to see this representation in the clinical trials. In one of the studies that we published almost 4 years ago now, we looked at the phase 3 trials of cancer drugs that supported FDA approval of these drugs, and we compared the patient population with that of the national representative sample by using the SEER database. And we saw that female patients were systematically underrepresented in the pivotal phase 3 trials, only 36% versus almost 50% in the general database. Black patients were underrepresented, only 2.1% versus 9.8% in the database. The patients with hepatitis B infections, hepatitis C virus infections, the patients with brain metastasis, they have been systematically underrepresented in the pivotal clinical trials. And there have been some efforts in this regard to broaden the eligibility criteria, to expand the eligibility criteria. ASCO has produced a statement saying that patients should not be systematically excluded based on their brain metastasis, or patients should not be systematically excluded based on their age group or HIV infection or renal function criteria, etc. But we still have a long way to go. So reading eligibility criteria is quite important. And this is important even for interpretation of the trial. I'll give you an example. The trial of sorafenib in advanced liver cancer. Sorafenib was the first targeted drug to show overall survival improvement in liver cancer versus placebo. And if you look at the pivotal trial, you see that patients receiving placebo had a median survival of almost 8 months, and sorafenib had a median survival of almost 11 months. So this was a 3 months improvement in survival, which looked all fine. But if you look at the real world, and there was this study done in the real world, in the real world, they found that patients who got sorafenib in the real world had a shorter survival than patients who got placebo in the trial. Just think about it. Patients getting placebo in the trial had better survival than patients getting sorafenib in the real world. Why does that happen? It is because of the eligibility criteria. The trial eligibility criteria is so strict that only patients with, we call them Olympian patients. They are patients with cancer, but they are so fit enough that they are eligible to participate in the trial. So, of course, their performance will be better and is not reflected of what happens in the real world. It's almost funny. These are patients with cancer in the liver, but one of the eligibility criteria was to have pristine liver function. And then after the eligibility criteria, we look into randomization, how the randomization was conducted. And, classically, this used to be a big deal, but nowadays almost all trials, they use computerized, standardized randomization system. So the randomization, I have never seen a problem with these big cancer drug trials. But the randomization ratio, classically, we used to see 1:1 randomization. But nowadays, we have seen unequal randomization. And that means randomization in the ratio of 2:1 or 3:1. And we do not have time to go through the details of why unequal randomization is not ideal. I would recommend people to read this article that I have cited here. But the case against unequal randomization is because an unequal randomized trial needs more sample size than a randomized trial. So we need more patients for such a trial. That means it will be more expensive to do such trials, and there will be less power to detect the effect size with unequal randomization. And that also means that because almost half of the phase 3 trials actually fail, we are unnecessarily exposing more patients to that unproven intervention without knowing that that is beneficial for the patients. And if we think that the interventional arm is so good that more patients should be randomized to it, then that means there is no clinical equipoise. And why are we even doing the trial then? And actually, there is no evidence that unequal randomization helps to increase recruitment into the trials. That's all for today's video. I hope you have found this useful. In the next video, we'll talk more details about methods. So we'll continue with the methods section. Thank you.